Get started. All right. Thanks again. Today, joining us are the Jumpstart class, Neurons Can Fly Jumpstart class students, and maybe some parents as well. And then on our side, we have Stacy Green here, the program manager, and myself, uh, Darwin Tsai, the principal investigator, and then uh, the lead of SciLab at the University of Michigan. I think at the beginning, I will start with some kind of general introduction slides, which I'll start to share my screen. I hope that you can see it over here. Let me move my screens around. Okay. All right, I hope that you can see the full screen. Give me a thumbs up if you see that.
Yep,
All right,
we
we're
can.
good. Okay, we're good to go. Okay, thanks again. Sorry for today. I think a lot of us wondered, because you're all high school students, a lot of us are wondering what college life would be. And especially when we advance from high school to college, after college, you may want to go to professional school, you may want to go to medical school, or go to a master's or PhD program. Um, but regardless, if you're interested in neuroscience or biology in general, you may want to experience what research is really like. So Neurons Can Fly is such a program that designed for high school student to experience what research in a cutting edge neuroscience lab really like, and give you the opportunity to participate in it. A brief introduction about myself. I am Darwin Tsai, Professor Crosby Kahn Collegial Professor of Cell and Developmental Biology at the University of Michigan Medical School. And I'm also a principal investigator of the Michigan Neuroscience Institute. My lab have these general research interests in mind. We're interested in how are the brain cells, in particular neurons, especially neurons, how are they generated? And then we're interested in, once the neurons gets generated, how are they wired to each other or connect to each other to form these so-called circuits, and then how the circuits work in terms of their function. And then we're also interested in the translational side of work. We're interested to discover where is autism in the brain, and are we able to treat depression? Some of the experimental pipelines or assays that we develop, and especially tools that we invented, are specifically designed to answer these questions. So my lab is a very interdisciplinary lab, which means that we deal with, on one hand, we deal with biology, chemistry, biochemistry, genetics. Those are very classically defined as, general biology fields. We also deal with optics, physics, um, microscope design, software design, so that we can acquire nanoscale brain structures. Also, we can use imaging method to record what is the molecules that are being expressed in the brain. Those molecules would be very critical to reveal the brain's circuit function, as well as the brain's neurochemistry functions. And those are related to the disease mechanism as well. And then we are utilizing these tools to also develop high throughput screening methods for drug discovery, for instance, phenotyping, and patient diagnostics. So those are the general interest of my lab. And then one of the model system that we use in the lab to do research, it's actually the fruit flies that you may sometimes discover in your kitchen or in your school, in your dining hall. So we kind of choose these seemingly harmless, but however pretty annoying little animals as our model system because it has lots of advantage in terms of using them for study. One important, uh, very important uh, similarity that the flies share with us is the genome, right? So you all learn from your biology class or from even from your middle school that the genetic material, the most important genetic materials in our bodies or shared by all the living organisms on Earth is DNA. The fly and human share very, very similar DNA. For instance, our genomes are over close to 90% similarity between us. And then in terms of gene-wise, fly pretty much have all the known genes that we have in our body. So they're called homologs or analogs in human and in fly. As a matter of fact, a lot of important genes are first discovered by the so-called mutagenesis screening on the Drosophila or on the fruit fly. The idea is that if you can randomly mutate, like here showed in red, some of the genome content in an animal, and you try to observe what is the defect, developmental defect, cellular defect, and then the functional defects of this animal. And then we can start to guess what a particular mutation can cause this animal misfunctioned. And then a lot of genetic tools and biochemistry tools, molecular biology tools have been discovered or developed to find out what exact mutation it is in this gene, and then so on and so forth, how does it affects the function. So you can imagine we can't really do this in human because it's not ethical. It's also would be very expensive to do it in larger animals, vertebrates. You may have heard about uh, lab mice or rats or other animals that have been utilized to perform biomedical studies. However, because how fast the fruit flies can uh, reproduce and how easy that we can create mutations and manipulate the genes in the genome, that makes fruit fly a very, very attractive model for biomedical research, especially in neuroscience research. So with the gene function being manipulated and discovered, the next, I already um, mentioned a little bit, is to try to determine or discover what is the origin of the pathology. Uh, and then we wanted to look at all the genes, let's say all the DNA or genomes in all the cells carries a particular mutation. However, it may not influence certain tissue, but only influence other tissue because how these genes are being regulated to express in the particular cell or a particular tissue. So that type of pathology determination is critical to understand certain disease, and then so that we would know which cell type or each tissue we should focus on to treat the disease, right? So one other thing I really like for using Drosophila for screening assays is that it can help us to test a lot of different hypotheses in an animal. What is it called? In vivo means in an animal. Because we can very precisely reproduce some sort of phenotype from a screening, and maybe multiple different mutations would lead to the same phenotype or pathology outcome. then we can introduce that purposely in the parent flies and to study how it can potentially be inherited through generations and what is the functions of one gene gets mutated versus two versus three, because a lot of effects sometimes it's a cumulative effect, may not be just an on and off. So that would give us a lot of way to test hypothesis much quicker. All right. So you can read a lot of these uh, texts here. Most of them I already touched upon uh, through the early introduction. But finally, I wanted to really also emphasize is the translational benefits. So because the similarity of the fly and human, especially uh, not only genome, but as well as its function. For instance, a fly can have dementia-like symptoms that they would be hard or easy to forget things or hard to train them to learn new tricks. That fly model can be used to screen drugs to see what can improve the memory function. So that's what is the translational benefit I'm talking about. And or there are ways that you can see that the flies uh, may express some sort of aggression or anxiety phenotypes. So they become a lot more aggressive when they see another fly. Normally, they don't really fight under plenty of food, but there are circuit mutations or biochemical mutations, pathway mutations that exist that we can uh, repeat some of the phenotype that we saw from depression patients. So that could give us some sort of develop a new assay for diagnostics. What kind of biochemical metabolites this Drosophila or fly will show differently than the normal fly if we mutate certain gene or manipulate certain circuit, and whether those metabolite will provide a new solution for us to do early diagnostic and seek for solution for intervention, right? So those are all the benefits of the fruit fly from a single tiny animal. That's why we love it. And then the next is that how are we going to study it? So here I'll show you one example that integrated some of the novel invention that my lab creates called the Brainbow, which is a transgenic technology to use to label the brain. So here show actually a micrograph of a fly brain. We dissected out from uh, this little animal here. So you see the red oval shape here represent the eyeball of the fly or the compound eye of the fly, which has all the neurons, and then the information processing layers uh, represent here to the two lobes of the optic lobe on the left hand side here, left and right sides here, and here in the middle, it's really what the central brain is an analogy to our human brain inside the skull. So in this case, what you're seeing here is many individual neurons. They're very tiny. Let me see what I can zoom in. Not really. Okay. So if you can see the little tiny circle here on Zoom, those are the cell bodies of the individual neurons. And you can see very long, like a silk thread like or uh, processes, we call them, or neurites. And these are the wires that connects between one neuron to another neurons in this animal. So the way that we label the brain, we will touch upon in some of the modules later in the Jumpstart class. The way that we label it is that we can express different color or dyes in individual neurons randomly, just like a painting that you would do in your art class. You would do color filling. So you fill the same object with the exact same color, but that object has a different color than its neighboring object. So here the objects are being individual neurons. then you can very easily follow their morphology, where do they travel, so you can guess uh, what these cells uh, communicate to other cells. How does that happen, right? So that's one kind of research topics in the lab is that identify different neurons and how do they wire to each other using this way. The other example of utilize the fly in showing here, you can see is you still see a lot of color. However, this case, individual colors do not represent a single cell. Instead, it represents a cluster of cells. Um, the zoom in here with the asterisk here, you will see a very big cell in the middle. Or maybe this one is easier to see. This green one has a very big circle here. That is actually if you heard about stem cells or the mother cells. And then next to the mother cell, we have a group of very tiny circles. And those are the neurons being born by these mother cells or stem cell. And then this wire or thread here represent the bundles of the neuronal processes for all of them from the same cluster. We also call it the progeny. So this mother cell divide, generate many, many daughter neurons or the whole progeny. And then each mother cell generate a different color, and then their progenies have the same color inherited. In this case, we can clearly segregate each stem cell, how many daughter cell does it generate, right? So, and then by introducing mutations, gene mutations that would disrupt this process, we would be able to know now whether that will cause a phenotype or that reduces the number of neuron that it generates, right? That will become some of the disease in human that have a smaller brain. And some of the mutation actually causes this mother cells or stem cell generate too many neurons. And that would cause a disease similar to a brain cancer. Not exactly, but similar to that, also have a inflated brain. So too many, not good. Too few, not good. We have to have the exact number of cells in the brain. So this kind of tool provides us provides an opportunity to use color to separate individual progenies. So now let me show you a movie to give a demonstration of one of the research projects that you may potentially have the opportunity to participate. In this movie, we show you a Drosophila brain, not just the central brain lobe, but also have the VNC. And these brains are labeled with different colors of neurons. And then we would use the software that developed by my lab um, so that we can reconstruct or trace them. And then we also develop AI tools to automatically generate these wiring diagrams. And by doing so, we can predict circuit function. We try to identify phenotypes with gene mutations. All right. Let me see. Okay. Okay. So now, for the next slides, I would try to tell you a little bit more about the camp itself or the program itself, Neurons Can Fly. So this program, my lab designed is for introducing high school students like you guys to neuroscience and beyond neuroscience, actually, many STEM related research. It depends on your interest and expertise. That we have integration of chemistry. It's all under the umbrella of neuroscience because neuroscience is very interdisciplinary. Physics, computer programming, AI, statistics, informatics, and then, of course, a lot of essays about behavior, animal behavior, and some of engineering design to how to automate this process of recording and analysis. Then our mission is to really the first goal is to inspire students to pursue higher education through the hands-on lab experience in neuroscience field or biomedical field, right? And then we also provide the opportunity for you to participate into cutting edge research. So this program is designed not like other summer programs or other extracurriculum programs that mostly just give you knowledge. That's not our goal. Our goal is to give you the proper amount of knowledge and understanding the research concept, right? To understand the research concept. And then potentially participate into the actual research project that's conducting currently going on in my lab, right? Being part of the research. So to do that, I already introduced the Drosophila fruit fly as our model organism. But the important thing is that when you finish the Jumpstart class of this online course, you would have the opportunity to do the wet lab and dry lab training in all these fields, right? Neuroanatomy, immunohistology, electrophysiology, microscopy, genetics, behavior analysis, image processing, neuroinformatics. A lot of words that you may not even heard before, but hopefully through the course of this 8-week course, that you would understand what these means and how does it link to the research that my lab conducts. All right. So our program has 3 modules. Right now, what you are getting into is this so-called the Jumpstart classes. So for the class, it's a self-paced class, which means that you will receive recorded lectures and also literature that you would write to read in your free time. And then we will pose open-ended questions for each modules so that it can build the knowledge for you to understand neuroscience in general, research in general, and then also very specifically cutting edge neurotechnologies, right? This is a requirement for you to finish in order to apply for the in-person summer camp that happens in July 2025, this summer. So the in-person summer camp will admit students. Once students apply for, finish the Jumpstart class, if you're still interested, you will fill up applications. Then we'll review the application based on the Q&A sections that you participate, based on the open-ended questions and how you answer it. We will select from the applicants to admit them into this week-long on campus at Ann Arbor, Michigan, this in-lab training, right? Here you will receive hands-on wet lab and computational neuroscience research training through interactive modules. So this is not a lot of lectures like which you already get prepared from the Jumpstart classes. Here is to really get to the experience of how research work, train you to conduct the day-to-day research practice that happens in my lab. The purpose of that, one is to, get you into it, get a taste of that, but also is to prepare for the third module of Neurons Can Fly program. It's called the Advanced Research Program. So this advanced research program will utilize the techniques, the skills that you learn from the in-person summer camp, right, you can apply that to continue on the actual research that happens in my lab. So if you are still interested, again, you will apply for another you fill up another application form, and then we will, based on your in-person camp performance and discussions, participation, so on and so forth, to decide whether you will be fitted into a particular research project of my lab. Each admitted student will participate remotely and or in person, depends on your geographical location, depends on the capability of the lab, that we can provide uh, the space and resource. And then you would participate research like that, and in addition, receive one-to-one mentorship in research analysis, data analysis, hypothesis forming, as well as writing, scientific writing, right? A lot of time, if you have a project that you might want it to convert that into some of your school science fair project or a state competition or national competition, and that happens in our past campus. So those are potentially possible to be integrated into this type of mentorship components of the advanced research program. Okay, so next I would hand over to Stacy, and who will give you a little bit more of a logistics details about the Jumpstart class and also the summer camp.
All right, thank you. Hi, everybody. So I'm Stacy. I'm the program manager, and I handle all the logistics of the program. So yeah, you all received an email from our Neurons Comply email account at some point this week, hopefully. And in that email, you were provided with the website that we're going to utilize for the Jumpstart classes. So if you haven't tried yet, please try to access that website and make sure that you're able to get onto it. This is what the page will look like. And the most important things for you all to be aware of are those yellow buttons that you see on the main page. So what Dr. Sai is pointing to right there, number one, the nervous system and anatomy. You'll click on that on the release date or after. Again, the idea here is that you're working at your own pace through the materials. However, our plan is to publish a new module each week. We have 8 total modules for an 8-week program. So it would be ideal if you could try to complete like 1 per week just to stay on pace with all of us. That will also help with the Q&A sessions, because the Q&A sessions are really going to be centered around the material from like the 2 weeks prior to that Q&A. So if you're caught up, that will be more beneficial for you. So you'll click on that yellow button and it will take you to the Google Drive folder where the materials will be published. If you click on that at this point, you're going to see that it's empty because we still have to put those materials in at some point today or tomorrow. Those will get released for you. So don't be alarmed if you click in there and you see it's empty. At this point, we just want to make sure that all those logistics are figured out and you're able to access the website and you're able to access the Google Drive folder. One other thing I want to mention about the materials is that you will not be able to make a copy of them or download them or print them. So you can view them on your computer, but just if you try to, make a copy or download it, you're going to get a notification that you're unable to do that. So that is set up on purpose like that.
These materials will be available until the beginning of the summer camp. So not just like the 8 weeks that you receive it, you want to come back and revisit it, and you will have the opportunity to revisit those.
Yes, and oh wait, can you still go back? Yep, so that second button right there, introductory materials, that's where we're going to put the recording of today's meeting. So that will be in that Google Drive folder. And then that last one you see there is just a shortcut. We will invite you to all of the Google Zoom meetings. You'll get a Google invite for those. but this is just another way that you can access our Q&A sessions. The dates are listed there. They're going to be on 4 different Saturdays from 1 to 3 p.m. Eastern time, 10 to 12 Pacific time. So you can click that button and join the Zoom meetings through there. So if you find that you are unable to access that website, please just send the Neurons Can Fly email address a message, and we will make sure that you get that straightened out. Okay, next slide. All right, so we do want to share just kind of briefly, if you decide you are interested and you are admitted to our summer camp that's going to be held the week of July 20th this summer, just kind of this general schedule and what you can expect. Again, the idea here is that when you come in person, we are really devoting the majority of the time to those hands-on activities. You'll already have all the prior knowledge that is necessary through completing the Jumpstart classes. So our daily day-to-day activities you'll see here are going to be a lot of like lab based activities. We're going to teach you how to use micropipettes and how to genotype our flies and do some brain dissections. We're going to do some fly behavior labs with our Drosophila, and then spend some time learning how to use AI tools to analyze the fly behavior. You'll get an opportunity to come to the Sci Lab and get a tour and see what an actual research lab at the University of Michigan looks like. So this is a really like fully packed schedule. We spend a lot of time during the day doing all those hands-on activities. And then we at night get to explore Ann Arbor, and we have fun activities planned with our counselors and with all of our students. You get to stay in a dorm right on the university campus, just like undergrads would, and you get to eat in the dining halls. So you get a little taste of what it's like to be an undergraduate student at a big university. Okay, so here's just some pictures from our past years, again, of our hands-on training modules. So you'll see like in that first picture, the students are learning some neuroanatomy using some sheep brains. In the second one, we have a student learning how to use our micropipettes. And in the third image there was taken from the microscope, just a normal compound microscope, where our students were working on trying to phenotype the flies by looking at their physical traits and trying to identify their various phenotypes. And then just some more pictures. We have when we do some genotyping, the students learned how to run a PCR reaction, so some results from their gels. Our optogenetic fly behavior lab, these flies are genetically mutated to where certain behaviors are determined by their exposure to red light. So when you shine the light on them, you'll see them carry out different behaviors. In this case, they would jump when the light was shined on them. So our students were doing that experiment and taking videos that they could then process later in a different module. We took that information and showed them how to process their videos and their images. All right, so again, kind of wrapping or following up with that AI analysis of fly behavior. In this next picture, we have a lab member that's working on the stereo microscope to actually dissect the fly brain from the entire organism. So you'll have some time to learn how to do that. And then we see Dr. Sai there with some students showing them around our lab spaces. So yeah, these are just more pictures. We could probably go through a lot of these, I think are repeated, but this one we have electrophysiology. Yeah, so then here at the end, some of the extracurricular activities that we do in the evenings after dinner, we schedule a tour of the big house. So the big house is where the University of Michigan football team plays. It's the largest stadium in the entire world. So that's pretty cool. We get to go down on the field and bring footballs and play around, and you get to go into the locker room and tour that space. We take our students on campus and we do some shopping, and they get to do things like go to one of our famous arcades called Pinball Pete's and have some fun. Last year, we planned some field games and did some friendly competition. We played some cornhole and different things just outside of the dorm just to get some fresh air. So that was fun. Okay, and then this is just a little map showing, no, it's okay, showing the campus area. So what you see over there on the left-hand side is the undergraduate science building. That is where we hold our summer program. It's in the same building, again, that our undergraduates would go to have their biology or their chemistry classes. So we'll spend the majority of our days in that building doing our hands-on activities. And then just across a bridge there, there's a walking bridge that will take you over to Alice Lloyd Hall. So that's the dormitory that the students will stay in. And then the dining hall is right next door. So students will have their breakfast, lunch, and dinner in the dining hall. And then you can see that big grass area right outside the dorm is what is called Palmer Field. And that's just a big grass area that's where we have our field day activities. And there's a track so kids can go out there and get some exercise and enjoy the beautiful weather that we have in Ann Arbor in the summertime. Okay, so I think that's it for my part. But you're muted.
All right. So those are the first two modules of Neurons Can Fly that will prepare you to a potential third module, which is the actual research part that participate in my lab beyond the summer program. Okay. So the continuing research options are for those who qualified and motivated, right? These are the two traits we really want to see you in the summer camp. The important thing is that, like I said earlier, each student can select a project. I'll pair them with a project that focuses on either on neuroinformatics or a systems neuroscience. I'll tell you a little bit detail of that. What does it mean? But just give you a brief taste of what we have currently. Our 5 students from 2024 summer program that are very engaged. So we basically pretty much right now, every 2 weeks, we will participate in a virtual meeting to do some neuroinformatics, kind of like we call it data mining process. And then two of those students are actually doing extremely well in that because of some of the new discovery that they had, it contributes significantly to the labs project that we actually included them as the co-authors on one of the newest publications that we will publish soon. So let me give you a very brief introduction to the first project for the advanced research program that is about neuronal or neural reconstruction in neuroinformatics. So basically, this is a purely remote project. So during the camp, in-person camp, you would learn the tools, how to remotely perform so-called the neural reconstruction. So here on the right-hand side, see these are the somas or the cell bodies of the neurons. You can see these processes. We utilize these labeling and images and kind of digitize it, trace them using AI tools. And then that reconstruction will be used to map to a atlas type of so-called flywire atlas, which is published recently on Nature, not by our lab, but by about more than 100 scientist team to spend more than 7 years to create this amazing electron microscope, electron microscopy based, imaging based diagrams that visualize pretty much every single neurons in the fly and the connections between them. However, because this is a static map, only showed you for similar to like the satellite images that you're taken from the air and then utilize that to create the US highway system map, right? So this is very similar to that. However, you don't really know that each city or each state or each part on that map, that when they connect to other places, what's the function of those particular cities or states, right? So in our case, because we have in the lab developed a lot of novel genetic tools to try to manipulate only these two neurons, for instance, their activity, and then look at their behavior outputs, right? So, but we need to somehow find a way to understand what is the input and what's the output of these neurons, for instance, up here. So then, because our new tool relies on those information and to specifically only manipulate these two neurons out of more than 100,000 neurons in the whole fly brain, so that we can understand these two specific function. So that is the informatics or anatomy-based hypothesis-driven practice that we would do. So use the platform that we developed, the neuroinformatics platform, and then the neural reconstruction platform to match the neurons from this brain to that brain to allow us to hypothesize what potential function these neurons can be and design experiments to validate those functions. Right? So the two students that I mentioned earlier, that they get acknowledged by the co-authorship on the latest paper of our lab is that they contribute very significantly in creating the protocol how to identify neurons from this image over here. Right. Once we have those hypotheses, that a lot of the other parts of the students will participate into more of a wet lab and behavioral neuroscience validation steps of the project, right? So these students will receive flies in tubes like this, will mail them to their home, and then they would start to do some experiments like this, right? Stacy just earlier mentioned. I'll let it play first, and I'll let you observe what happened to these flies. Okay, so this is an experiment or result that's submitted by one of the Neurons Can Fly student. So what you have seen there is that when we turn on the red light, this fly jumped. Hold on, let's go there, right? It creates a sudden action of the fly that amazingly, there are only about 10 neurons being stimulated by the light, and then the fly's action respond to those 10 neurons. So the idea for us is that we have quite a few of interesting circuits that we wanted to discover further to understand what their normal function is and what their function when it encounters a disease model in a disease model. So then from the first project, we will identify potential neurons that may participate different functions, and then we'll guide the students to generate transgenic new transgenic animals. Then they will perform these type of experiments, and then do the analysis, right? Use AI to help to annotate the images or the videos, and then we will need to perform statistics to figure out what are the potential defects of flies that have disease or just a normal function. Right, so this is called the jumping behavior to show you, but we have many other behavior we wanted to explore. So that is the second program. So now and that would also conclude my today's presentation for this Neurons Can Fly introduction. I will be happy to take questions if you have any. You can just, speak up or raise your hands or type them down in the chat if you have questions. there's a question in the chat, where's the Jumpstart class located? So the Jumpstart class is completely remote, right? Like we said earlier, we will send out these informations and then through the email communication, and then also the landing page that Stacy showed you earlier, you all receive it on the onboarding email. Those are the entrance. And then it's a self-paced class, and then we will have Q&A sessions every other Saturday.
So yes, I would recommend that you bookmark that website because all of the classes will be accessed through that website in your onboarding email, which you should have already received.
Yeah. You should have received that website link already. If you still don't have an email, just email neuronscanfly.umich.edu. Will both the advanced research project be available remotely? Both of them are designed to be done remotely, yes. So another question, how will we submit the assignments such as the essays? Those will be filled up Google Forms. In the next question will help the students to write the scientific paper after advanced research if it is possible. If the students motivated enough to write their own essay, we will provide some help, guidance to that for sure. The next question, will we have any quizzes, tests, or checkpoints to ensure we are learning? Yes, so those are the questions that will be after each module, you have the materials, there will be some questions related to that. In terms of quizzes and tests, and these are not really like the standard knowledge consumption classes like you have in high school or college, the questions that we put in there, you would need to use the knowledge that you learn from there, and also potentially you would Google by yourself to fill up the questions. If so, those are the way that we examine students, not a standardized test. So will the form be in the weekly module? Yes, the forms will be in the weekly modules. Yes. Next question, how will the Q&A session be structured? So it will be a Zoom meeting similar to what we have today. So through the Zoom meeting, because the forms that the students filled up, we will provide insights and analysis to what the students' question have, and then our own kind of like not saying answer, because these are all very open-ended question, right? So this is very different than your quiz and tests that you're very familiar with. What we really have is that in the Q&A, we will guide a discussion of particular paper or questions. We'll pick from there and analyze what the students most response to the question, will design that and lead the Q&As. So the next question, could you please repeat the emails that I should send to even have that in the link? I'll type that in the chat right after answering these questions. Okay, next question. Will the students use university lab for advanced research topic? So there are in-person possibility that student can come into the lab for those exceptionally good and mature student, because after all, you guys are all minors. So we need to take some responsibility, but you need to go through kind of background check as well as safety trainings, online trainings. And then for those exceptional students, we will provide the opportunity for the student to come to the lab and stay in the lab to do research inside the lab. So those topics will even be beyond what you have for the current designed more of a remote modules for the advanced research, continuing research. Okay, so the next question, are these set due dates, or can we work on Jumpstart mostly at our own pace? yes, you can work on your own pace completely. The Q&A sessions are really to inspire you to discuss, to think critically, like a scientist. It's not a knowledge exam, so no pressure on the exam at all, right? What I'm looking for to provide the training opportunity or inspiration is that you will participate in a very different type of learning experience. That is how research is conducted based on the knowledge that we know already. The critical thing that advances human society is not that how well that we can repeat the previous person did. That is that can we jump out of the box to present hypotheses and testable hypotheses and design experiments to test them and to validate, right? So that is the focus, even for the Jumpstart class. So that's very self-paced. And because of this nature, if you missed any Q&A sessions, you can always participate by watching the videos that we post after. Will there be due dates on the forms like they being due on the week they are assigned? So that's again, there's no due dates on that, but we will only discuss the Q&A or questions centered around the students already filled the forms. You are always welcome to compare notes afterwards, right? This is completely self-paced. Next question, do you have any website or resource that you recommend? Additionally, is there a certain subject you recommend we learn that would aid in our learning of neuroscience? We will provide, in addition to the video that I will post later today, for the first module, you will see how it works. We'll also provide some recommended textbook reading, scientific article reading, so on and so forth. So that can help you to gain a broader knowledge beyond what I would say is the minimal required knowledge that you need to participate for the Jumpstart, for the summer camp. So next question, when will the application for the second phase be and when the decision will be made since the phase 1 study will last till mid-June? That is a great question. So the application can fill in when you don't have to finish the whole thing, right? There is a rolling admission that we would do. Similar to the Jumpstart class, you can start this week or later weeks, that's fine. But your application and the participation through this whole Q&A process and through the Jumpstart process give us enough data point that when we feel that comfortable that this student we wanted to admit, we will send out admission. I know that sometimes it's a little bit of, could be a conflict of what you may wanted to feel that week with other activity. But in this case, it's a bi-directional selection process, and it's very much like a college admission to a certain degree, although we have a much shorter announcement time. But we will try to, once we decide a student is suitable, or once you determine that you wanted to participate, please apply, and we will make that decision, and then you will have time to decide whether you wanted to participate. So the next question is, will the application for the summer program have an essay or short answer portion? Yes, there will be a short essay portion to it. Next one, will we get personal feedback on our submission on the form? If so, how? This will potentially be difficult for the submission form. So I think if you are really eager to know why it's not being able to admit it to the in-person summer camp, uh, we may potentially let you know, but sometimes it's a confidential it's a confidential issue that we do not want to disclose some of the information. People have to compare between students. And then also a lot of time it's not because you're not good enough, it's just because we don't have enough space. Currently, we are aiming for providing about 25-ish positions, because the resource of the university and then the resource of the lab is limited. So a lot of time that also because it's a first come, first serve. If we cannot really decide which student to go, then we have to admit someone that's really eager to do that, the motivation. Next question, do you guys code programs for modeling? If so, what language are they coded in? Language we use are Python. The most language. We use lots of languages, C, C++, Java, JavaScript, Python. But for you guys, if really interested in informatics and data mining, Python would be the program to do. Okay, next question. How many students can attend summer camp? What is the cap number? I just mentioned somewhere between 20-25-ish. Okay, next one. If I applied but wasn't picked for the in-person camp, would I be able to apply next year without taking the Jumpstart class again? Yes, the answer is yes. If you apply this year, next year you just need to fill up your application again, because you can learn, you're more matured, and you learn more about backgrounds, and you know more about research, so on and so forth. You're still interested, or because of the time conflict that you cannot participate this year, it's open for you to apply for next year without taking the class again. The next one, if the student only be interested in one area, science or computer, would the student still be attending this program? Yeah, still can attend this program, of course. The two projects at the end, even though even for someone, so first of all, if you are really interested in because of the advanced research program, you still are required to participate in the in-person camp, even you are purely a computer geek. So the reason why that is because we are applied science, right? We're not pure math or pure physics. Those are theoretical sciences. We are still applied science. In this case, even you are a coder, you wanted to know how your data is generated, because there is a saying in our field is garbage in, garbage out. If you do not know or control what your data source is, you cannot communicate with your teammates, your colleagues that provides the data to you. You don't understand how that actually created, and you would not be able to understand what statistical tools or modeling tools that you wanted to apply to do the research. So that's the reason why all the students, regardless it's project one or project two interested, we still need to participate at the in-person summer camp in order to move forward. Okay, I have one more question here. Do you have any office hour so that if we have questions, we can join the call and ask them, or do you prefer we message you? So you can always email, right? Neurons can fly. And then during this jump start class, and then the Q&A session is the office hour, kind of like the office hour. You are prepared, and you're very welcome to discuss the questions that we leave there, but that's sorry, my office light. But you are also very welcome to ask any questions that you have through the class beyond the question that we gave to you. Okay, so let me copy the email address for you. The Neurons can fly.
Darwin, if you also, Oh, I was gonna say, if
Mhm.
you also have it accessible, do you want to put the website link in the chat as well, just so
Yeah,
everybody
that's in
has
there.
that? Okay,
That's
thank
right.
you.
Just give me 1 second. I just wanted to make sure. Okay, so in the chat, I am about to type in the website of Neurons Can Fly, and as well as the email address. Hold on, give me 1 second. Copy email address. I'll copy that to the link. Okay. So yeah, those are the two websites. Yeah, and then the email that you can ask questions and get more information. Okay. Is there any other questions? Okay. If no, thank you very much for attending today's meeting. I'm looking forward to seeing you on Zoom and discussion about science and research. All right. Thank you. Bye.